with acquired immunity. So we finished off on this slide, and we said, yay, MHC and CD4 are interacting, so MHC2 and CD4. Our other combo was MHC1 and CD8. So what does the CD4 and MHC interaction tell us? If you were CD4, what's your main purpose in this immune cell in reaction? You slept since then? I'm shocked you have.
It helps it bind, right?
What was that binding tell us?
That's the cell bind. Don't know that much.
Then it's our own MHC doing the presentation. So CD4 is telling us that it's our own MHC2, CD8 is saying it's our own MHC1. Then the actual T cell receptor is what's binding to that epitope. So B cells, on the other hand, don't need stuff presented to them on any MHC. They can bind and recognize an antigen just directly. So if they have their B cell receptor hanging out on their surface, it can bind directly to its specific epitope that's going to be like something specific on the surface of E. coli or staph or anything like that. As it does that, then the B cell receptor binds whatever specific microbe or virus or toxin or whatever it's going to bind. That binding is going to cause that thing to be internalized. It's a nice strong binding. Even if, say, it's a motile bacterium trying to swim away, it's not going to be able to if that B cell receptor hasn't latched onto it. So now whatever it is gets internalized into the B cell. Now, just like when we talked about macrophages and dendritic cells being a professional antigen presenting cell, it's going to chew up that microbe, isn't going to spit out the fragments, instead it's going to chop them into these 10 to 14 amino acid segments, present them out on MHC2. Remember, MHC2 is presenting stuff acquired exogenously from outside the cell. So now the B cell, acting as an antigen presenting cell, interacts with CD4 and the T cell receptor on this T helper cell that we just talked about, it getting activated here. So T helper cell is going to be the in-between one in this. It has to be activated to then stimulate the B cell that's seeing the same pathogen. So say E. coli is infecting you. T cell receptor interacts with the antigen presenting cell to recognize, oh, it should be activated. This T helper cell is activated. B cell comes across that's also specific to E. coli. It's going to take it up, also put it on MHC2. Now this T helper cell says, oh yeah, what you're presenting is the same thing I just got activated again. Let me help you out. So now B cell is going to interact with this T helper cell at this receptor complex, this T helper cell that just got activated, which may have been through another B cell or a macrophage or a dendritic cell, any professional antigen presenting cell. Now it says, yes, I know how to fight this. I just got the signal on how to do that. Let me tell you how to do it. And this interaction happens the same way it did with this T helper cell when it got activated. So now, chemical mediators, receptor recognition, this B cell is going to be activated. So this interaction again is now going to activate the B cell. When the B cell gets genetically activated, it's going to do a couple things. It's going to increase in size. It says, yeah, I know how to do things. I'm going to get bigger now. It's going to start synthesizing proteins, and the main protein it is going to synthesize is a whole bunch of antibodies. So B cell is going to start multiplying through regular cell division, make more copies of itself. They're exactly identical, so this is clonal expansion. This B cell is only specific to whatever pathogen its B cell receptor recognizes. So this got activated because it's specific for E. coli. This B cell that's expanding and now making lots and lots of copies of B cells, they're also just specific to E. coli. This won't help against COVID or the flu or Ebola or any other pathogen. So this was on this side here. And as this is happening, most of our B cells are going to become what are called plasma cells. Plasma cells are going to be the ones that are meant for the immediate response. They're going to make a whole bunch of antibodies that are for the current infection happening. The other batch of these B cells, a small fraction, maybe a fourth, an eighth, or a tenth, somewhere in that range of these activated B cells, they divide, but a smaller fraction of them will become memory cells. They're not going to help during the immediate reaction or infection, but they will help at a future time if you get exposed to E. coli again in the future. So memory cells are going to be long-lived. They get reactivated the next time you see the pathogen. They'll have no effect this first time when you're currently infected. It's only the second time around. They do continue multiplying after committing to memory. So say this cell replicates, you make 10 cells, maybe 9 of them become plasma cells that are making a whole bunch of antibodies for that immediate response. One of them becomes a memory cell. This memory cell, once it's committed to becoming a memory cell, it's going to continue dividing. You don't want to put all your eggs in that one memory cell basket. You want to have a few options. Most of these activated B cells become these plasma cells. So plasma cells are huge cells. They're terminally differentiated. So this plasma cell could not decide, hey, I'm done secreting antibodies, I want to become a memory cell. It doesn't have that option. Once it's a plasma cell, it's always a plasma cell. These are short-lived. Like usually they'll just live a couple days. But in that time, all they're doing is spitting out never-ending amounts of antibodies. And they're specific to their specific antigen. So if this got activated against E. coli, all it's doing is spitting out E. coli specific antibodies. And often they never leave the secondary tissue. So they might be hanging out in the lymph nodes, like under your armpits, in your groin, under your neck. That's where they'll hang out. And when your lymph nodes actually swell up, like when you're sick and you feel them swelled up, or you can obviously see that they're swelled up, that's because all these B cells have been replicating nonstop. And T cells for that matter also. So that's what's actually causing the swelling. And then they'll remain in those lymph nodes, but secrete the antibodies from there. Since we know that lymphatics and blood circulation are connected, then they get secreted into the lymphatics, they'll go then into the bloodstream. Antibodies, and we kind of talked about their basic structure earlier, but now we get to go into more detail. So they do have a basic structure. They have the antigen binding fragments, or Fab regions. There's an amino terminal, just like any protein has. So that's on one end. And that has a groove that's going to be formed by our variable region of our light and heavy chains. So if you remember that Y shape of an antibody, at that two ends, each one of those is a binding region for its specific epitope. And it will, again, only bind one specific epitope. So that means one antibody can bind two separate epitopes. The other part of an antibody is going to be the crystallizable fragment, or FC region. That's the constant region. It's going to be interacting with other immune cells. So it's the thing that if you coat some pathogen, say with a whole bunch of antibodies, then other phagocytic cells like neutrophils could come along and say, hey, this is coated with antibodies, I'm supposed to destroy it. So, going through some videos. First is clonal selection. So once we've activated a specific B cell especially, how does it expand against that pathogen?
The process of B cell activation is called clonal selection. During this response, a B cell with a specific antigen receptor recognizes an antigen. This cell proliferates to produce a multitude of cells, also called a clone of cells, with the same specificity, which differentiate into memory B cells and plasma cells. This process is called clonal expansion. The antibodies produced by the plasma cells bind to antigens to produce the humoral response. In the simplest form of B-cell activation, an antigen called a T-independent antigen enters the lymphatic tissue and binds to a B-cell receptor, or BCR, on the surface of the B-cell. The B-cell receptors on a particular B-cell are identical and very specific to just one antigen. Once the BCR binds to the T-independent antigen, the B-cell becomes activated. It's now ready to proliferate. However, most antigens are not potent enough to activate B-cells by themselves. In these cases, the B-cell will require a second opinion from a helper T-cell in order to become activated. We call these antigens T-dependent antigens. The helper T-cell recognizes the antigen as it's presented by the B-cell and secretes cytokines to tell the B-cell to become activated. Once a B-cell becomes activated, it proliferates or divides, forming a clone of cells specific to the same antigen. While some of these B-cells differentiate into long-lived memory B-cells, most differentiate into plasma cells, which secrete antibodies against the antigen that activated them. The antibodies can enter the bloodstream and travel throughout the body. Plasma cells only live for a few days, but each one can produce about 2,000 antibodies every second, resulting in enormous quantities of circulating antibodies.
And very rarely do you actually have the T-independent reaction, just like the video said. Most of the time, you need that second opinion. You need that T helper cell to say, hey, I already saw this thing, I know how to fight it, let me instruct you how to do it. So, antibodies themselves, they have a lot of function. And that's why, oh yeah, get vaccinated. That's one of the reasons is because you're going to have B cells that produce antibodies. So an antibody can serve a lot of different roles if you're exposed to their reactive pathogen or virulence factor. So it doesn't have to be an intact bacterium or virus. It could just be a toxin or some other specific protein that's being secreted by some pathogen. So some of the things antibodies can do. You can go through opsonization, so that's when you're going to coat the antigen with a whole bunch of antibodies, and that makes it easier for phagocytic cells like neutrophils and macrophages and dendritic cells to swallow us. Like say there's something that makes a capsule, we know capsules are anti-phagocytic, but if you have antibodies that bind to that capsule, now a phagocytic cell can wrap around it and says, hey, I know how to eat this thing now. So in this case, you'll have receptors on the phagocytic cells that specifically bind to that constant region on the antibody. You can also go through agglutination. So that's when you're going to actually just physically tether pathogens together. So this is whole cells, like whole bacteria, and this can make them immobile. Even if they have flagella and trying to swim away, they can't because they're tied together with a Y-shaped antibody. Precipitation is going to be similar clumping like what you go through with agglutination, but it's going to be of just small free soluble antigen molecules. So agglutination, whole cells, precipitation, soluble molecules. But if you've gone through chem, like Chem 20 or something, you've seen precipitation reaction. Something that was soluble, when it binds up like this, precipitates out of solution. And that's going to make it easier to filter by things like your kidneys and your spleen. You can also go through a process called complement fixation. Complement I'm steering away from in this class. If you ever get to take an immunology class, you'll hear lots about it. But it's a series of proteins. You always have it floating around in your bloodstream. But they work in a specific series, and one will bind, then the next one binds after that, then the next one after that. So you hear things like C3, C4, C5, and so on. Those are the individual complement proteins that have to bind in a specific order. But once they all bind, they're going to form a drill, and that drill is going to punch holes into these cells. So in this case, antibodies, they have a site for complement proteins to bind to. And then once one binds, then the next one binds, and the next one binds. So having antibodies already bound to some pathogen make it easier for that drill bit to sit down and poke a hole in the whatever the pathogen is. So another thing it can do is just neutralization. If it coats the whole thing, that thing cannot bind. So antibodies can do that to the surface of a virus. We remember how viruses work, they usually bind to some specific receptor on our host cells, and that's through those spikes that they have on their surface. But if you coat the whole virus, that spike can't bind to that receptor. Same thing with toxins, like say some toxin produced by a bacterium. Usually those toxins bind to specific receptors, like when we talked about AB toxins. So now if that binding part of the toxin is coated in antibody, it can bind to the cell receptor. You prevent that interaction and they're completely inactive. So a lot of times you get, say, you get bit by a rattlesnake, you get anti-venom, and all anti-venom is a whole bunch of antibodies against that venom. And it does the same thing, you just get that treatment with the anti-venom, like and by IV, and all it's meant to do is go throughout your body and bind up all that rattlesnake poison, just by coating it in antibodies. Same thing works with various bacterial exotoxins the same way. But as I'm sure you've noticed, antibodies are going to be mainly useful in combating pathogens that are actually outside the body or outside the cells. Same thing with toxins, but things that are outside in, say, the blood, the lymphatics, the extracellular fluid. So things like viruses, bacteria, protozoa, the worms or helminths. But antibodies are not going to help if something is already inside a cell. Like when a virus takes over a host cell, antibodies are going to be completely useless to combat those once they've already taken over the cell. Because antibodies can't go through the plasma membrane of your host cell to bind up anything inside. So going through all our things we just covered, you can just coat something with the antibodies to say, hey, do something to me because I'm bad. Opsonization is going to make it easier for pathogens to be taken up by phagocytic cells like macrophages and neutrophils. Neutralization is going to be when something like a virus or a toxin is coated in antibodies so that now it can bind a specific receptor on your host cells. Agglutination is going to be when you're going to physically cross-link some type of whole cells, like whole bacterial cells. Say these were two E. coli with happy little flagella swimming around. One of these Y-shaped antibodies binds on one and binds on the other. Now neither of these can swim away because they're both bound by this antibody. Complement fixation, then something gets bound up by antibodies like this. Then on this FC region, that's where there's the complement binding site that allows complement to sit down, and then it's going to poke holes in the cell membrane. And lastly, precipitation, where we are going to have soluble molecules, like maybe a toxin or something floating around in your blood or your lymphatics. It binds it up and then causes it to come out of solution. Not just one type of antibody exists either. There's multiple kinds out there, and they all have their own specific purpose depending on what type of infection you have and where it's actually located. So there's 5 different basic classes called isotypes. And a lot of times if you're checking for an infection, the doctor would not just say, what are your antibody levels, but then they'll look at specific types of antibodies. IgG is the most common of those 5 isotypes. It's a monomer, meaning it's just one Y-shaped protein. And it's going to be produced by your plasma cells during your primary response, your first response ever to some pathogen, and then it's also going to be produced by your memory cells during a secondary response. And last slide we'll cover is this primary versus the secondary response. So IgG is going to be your most common isotype in the blood, the lymph, and the extracellular fluid. It's also the only isotype that can actually cross the placenta. So while the baby is developing in you, this is the only one that can actually go across that umbilical cord and then actually protect from an infection while the baby is still developing. IgA, so it's going to be a monomer in the blood, but it's actually a dimer at mucous membranes, places like along your intestinal tract or your respiratory tract. So that's where it's especially going to be useful. Also along your genitourinary tract. It's two Y-shaped structures held together where the two Y's are sticking out. So that means one of these IgAs as a dimer has 4 antigen binding sites. So it could bind up 4 different things at the same time. So it's great for providing local immunity. In some cases, non-specific, like you'll have IgA that binds to peptidoglycan along your intestinal tract, and that helps to just bind bacteria just for kicks. IgM, it has even more potential. So it's a pentamer. 5 individual Y-shaped antibodies all held together like a snowflake shape. Eric, was that a yawn I saw?
No, I'm just stretching my jaw.
Too much chewing during the day, kind of working out the tension. Yeah. It couldn't be yawning at antibodies. I mean, that's a direct insult to your own immune system.
It's super. It's like war between your body and bacteria. Cool.
Right? Exactly.
It's impossible for this stuff.
So, because it has 2 antigen binding sites per antibody, and there's 5 of these all attached to each other, then you can bind 10 of the same antigen at the same time. So that's why it's often the first isotype that you'll produce in any infection, viral or bacterial or protozoan or whatever infection you have. Often you'll go through producing a whole bunch of IgM first because you just want to bind up as much of the pathogen as you can early on. IgD, so it's a monomer, but it's found in very low levels in your serum because it's the main thing that serves as your B cell receptor. So the only time you would actually find it in solution is if a B cell dies and then releases that receptor. But most of the time, it's never secreted by a B cell. It's just attached to the surface of the B cell. And it's the one that actually can bind directly to the specific epitope and trigger internalization, chewing something up, and then presenting a fragment on MHC2. IgE, the last version, so it's going to be a monomer, and it's going to be what we'll talk about on Thursday when we cover allergies. It's the one that causes most of your symptoms of runny nose and itchiness and watery eyes and all of those things that you have when you have an allergic reaction. That's going to be due to IgE. Also big for helminth infections. If you ever travel to Asia or parts of Central America or somewhere and pick up a worm infection, if you don't know that you have a worm infection, your immune system will start to fight it anyway, and that's because it's going to produce a whole bunch of IgE. There's that constant region of IgE, and that's going to be great when you have a worm infection, but bad when you have an allergy. So cells like mast cells and basophils, they love to secrete things by degranulating. And in the process, as we'll talk about in more detail on Thursday, they're going to release a whole bunch of histamine, one of the things they release. And that histamine is the main thing responsible for a lot of your allergic symptoms, like your runny nose and watery eyes. And that's because they bind this Fc region of antibodies. So IgE is going to also recruit other immune cells to the site of an infection or injury, like a worm infection, normal allergies, asthma, to the extreme level anaphylaxis where you can dive, like if you have a peanut allergy or a shellfish allergy or something, and you suddenly go into shock within a few minutes. Usually carry around an EpiPen. That's because you can go all the way to anaphylaxis, all thanks to IgE. Lastly, types of responses when we come to antibody responses. So you have the primary response and the secondary response. Your primary response is going to be the immune response to the first time you're seeing an antigen, whether it's through vaccination, just natural infection, first time you're being exposed to something that you're going to be allergic to. So early phase of the primary response is called the latent period, and that's when you're still starting to make antibodies. But you've never seen this before, so it's gonna take a while. You have no memory response to it. So you have to go through all the parts of antigen presentation, finding those few specific B cells in your lymph nodes that actually recognize it. They have to get making proteins, then they'll start secreting antibodies. Have to concentrate the antigen itself, activate those. So it takes a few days of that, usually 10 to 12 days to get that primary response to actually occur. And of course, during that time, your innate system's in action, but at the same time, if that can't contain it, you could totally die in that 10 days or 12 days until you get your adaptive site going. And the same is true for T cells. They also take about that long to get activated the first time you're seeing a pathogen. Second time you see it, called the secondary response, that or anytime after second, so third, fourth, fifth, all of that counts as a secondary response. And that's purely going to be dependent on your memory B cells this time. So now you've already seen it, you have those memory cells stashed away. They just need to say, oh yeah, I remember when I came across this like 10 years ago, I know what to do really quick. So now you don't need to go through crazy amounts of time to get these activated. This response should also be stronger, faster, and longer. So because you've already seen this before, you know exactly what to do. So you're going to have more of these B cells made. They're going to last longer in producing antibodies, and they'll do it in a much shorter amount of time because they're already there waiting to be reactivated. So if you were to compare the two responses, primary response, you have this latent period of 10 to 12 days where you would measure no antibodies whatsoever. Infection still progressing along, but it just takes a while to, say, activate your specific B cells and T cells against this pathogen. Then, first response of your primary response is going to be to produce IgM, that pentamer, because you want to bind up as much of the pathogen or toxin as you can the first time around. And then you finally start producing IgG or IgA or IgE, depending on where the infection is or what the stimulant is. This could apply to, say, an allergen the first time you see it. If you're going to be allergic to peanuts or shellfish or a wasp sting or something, the first time you're stung by a wasp, you'll go through the same process as if you were infected by a bacterium. And you can see the number of days like this. So 20 to 30, that period is when you have your highest antibody level. Now your second time you're exposed, things happen very quickly. So IgM is still produced, similar levels to bind up stuff, but it happens right away. Within the first day, you're already starting to produce some. And more importantly, now you're producing IgN, IgG, IgA, or IgE, but you're already way up here by day 5, versus you didn't hit that level until day 25, the first time you were exposed to it. And this is because now your memory B cells that you had stashed away are converting to become plasma cells again. So some of them are going to replicate, and some will automatically become plasma cells. And again, as soon as you become a plasma cell, your only function then is to produce lots and lots of antibodies. So during this time, your rate of production, your levels of production, obviously way up here, and how long they persist. So you're well beyond day 30, and your levels are hardly dropping. This is some of the stuff they're trying to figure out with like the different COVID vaccines. How long do your antibody levels stay elevated after you've actually gotten the vaccine? And that's this response here is also called the anamnestic response. Basically the secondary response. How strong is that response? So, also relating to those B cells, the other thing that of course has to be active are the T cell responses. We know the T helper cells have to be involved, especially that first time, and again the second time, to activate those B cells. Have to say, hey, I know, I've seen this, I need to tell you how to do it. So they also are being activated. you hear all about the antibody response, like when they look at how effective a vaccine is, but T cells are somewhat the big part of that also. The T cell immune response is also called the cell-mediated response, and that's because it's the actual cells doing the responding. Unlike the B cells, the humoral response, it's the antibodies that are being secreted by the B cells that are having the effect, versus the T cells are actually, they themselves are involved. So things like killing infected host cells, a B cell can't do anything like that. Only a T cell can do that. Remember, T cells have to interact with antigens, either presented on MHC1 or MHC2 to actually be activated. And you're going to have different classes of T cells that are going to have different functions. That means they're going to produce different sets of chemicals, like cytokines, that are going to have different effects. So if a T helper cell secretes certain cytokines that might be against an intracellular infection like a virus, secretes a different set of cytokines, it might be against, say, a worm infection or some other parasite. So cytokines are just substances that have an effect on other immune cells. And they're a subgroup of chemokines, which are any chemicals that have an effect on another cell type. T cells can modulate the activity of other T cells, antigen-presenting cells, and B cells. So, some of this may be reviewed from previous slides, but it leads up to our final type of cell, which is the cytotoxic T cell. So, two types. We have the T helper cell, which has CD4 on its surface, which we know is there to see is it my own MHC2 doing the presentation. And you have the cytotoxic T cells. They have CD8 on their surface to say is it my own MHC1 doing the presentation. Both of these are still going to be stimulated by MHC peptide complex. T helper cell, you have the T cell receptor. It binds to some peptide being presented on MHC, specifically MHC2, and then the CD4 on the surface of this T helper cell is going to recognize that it's my own MHC doing that presentation. And we'll go into that on Thursday when we get into like organ transplants, and why those often fail is because it's not your own MHC doing the presentation. Cytotoxic T cells, on the other hand, which we haven't really talked about yet, they have the T cell receptor on their surface. Now it's going to bind to the peptide, usually 8 to 11 amino acids in length, and then its CD8 on its surface recognizes that it's your own MHC1 doing the presentation. So now, either way, T cells get activated, they mature, and they divide by mitosis. And just like with the B cells, some of these T cells are going to become memory T cells, which can again protect you years or even decades down the line. So, same thing on the surface of these also. You'll have CD4 on the T helper memory cells, and you'll have CD8 on the T cytotoxic or cytotoxic T cells. So now if we put all of this response together, say you have these CD4 positive T helper cells. They're the puppet masters. They're the ones that are pulling the strings, because they can regulate how strong your B cell response will be and how strong your cytotoxic T cell response will be. So we already know they're important for your activating your B cells during those T-dependent responses. You won't get a lot of antibodies produced unless you have the T helper cell already saying, yes, I've seen this pathogen, let me help you by giving you a bunch of cytokines to say how to respond. T helper cells can also activate macrophages for increased phagocytosis and killing, and that's going to be when you have an intracellular infection, like a virus of some kind, or an intracellular bacterium that said, yeah, I want to hang out in the lysosome of this cell. So this is when it can say, hey, you may want to do something about this intracellular infection, maybe destroy a lot more stuff that you have inside of you. So cytokines are initially secreted by an antigen-presenting cell. That's going to dictate then the specific cytokines that get secreted by the T helper cell. So this initial response here, this will be, say, a dendritic cell, a macrophage, a B cell. any of those as professional antigen presenting cells, they come across a T helper cell. So same way T helper cells can respond, they have to bind to MHC2 with some epitope. CD4 has to recognize that MHC2 as being your own. Now you can have a couple of different types of T helper responses based on this initial interaction. So based on what it senses is happening from this antigen presenting cell, is going to dictate what kind of response it says for other immune cells to have. T helper 2 responses are the ones that are for extracellular responses. You want to signal B cells to make a whole bunch of antibodies because there's some extracellular bacterium or a worm or something in the body. So it's going to release specific cytokines like IL-4, IL-5, IL-6, IL-10. All of these are to activate B cells because you want mainly an antibody response to something extracellular. T helper 1 response, on the other hand, is if this T helper cell realizes based on this signaling that, oh, whatever the pathogen is an intracellular thing, like a virus that's already infected cells, an intracellular bacterium, maybe a cell has become cancerous and cancer is starting to spread throughout the body. So in that case, producing antibodies is useless. The problem is inside the cell. So in that case, in a T helper 1 response, you'll produce TNF, interferon gamma, a few other cytokines that are going to be specific for intracellular killing. So that's going to stimulate macrophages to kill what's inside them and to actually go through more phagocytosis of other stuff also. And of course, this is the simplified version. In reality, usually it's a mix of, like, depending on the type of infection, like a viral infection, the virus comes out of the cell, you'll want a T helper 2, and when it's inside the cell, you want a T helper 1. So often it's like so much percentage T helper 2, so much percentage T helper 1. There's also other types of responses like T helper 17, T helper 22, all these different types of responses. Ever taken regular immunology class just for kicks and grins, feel free to hear all about this. So the CD8 positive cytotoxic T cells, once they get activated, they're going to kill other cells that are targeted. So like virally infected cells or cancer cells, only thing that can work against those are going to be the cytotoxic T cell. So cytotoxic T cell gets activated initially, so it's come across its specific epitope being presented on MHC1. It gets activated. Now it's going to say, hey, okay, I got a mugshot of whoever I'm supposed to kill because I'm the assassin. I'm going to go around the body patrolling looking for that mugshot, that picture of its target. So it's going to sample a whole bunch of cells. And anytime it samples MHC1 and sees some other epitope, it's just going to ignore it. Remember, all cells besides red blood cells express things on MHC1. So cytotoxic T cell that's activated, it'll keep searching around. If it's not its target epitope, it passes it by. But when it gets to its target epitope then, so now it's gonna say, hey, I'm gonna kill you. But it won't even give it much warning. It's not even gonna say anything, it's just gonna kill it. And when it does that, it's gonna use 2 types of enzymes, perforins and granzymes. So the perforins are there to perforate. So they get secreted first, and they're going to poke holes in this cell. This is the target cell that's, say, infected with a virus. Could be a cancerous cell also. Could be a cell that has some intracellular bacterium that's replicating in there. Remember, MHC1 is just there to say, here's what I'm making internally. So this cytotoxic T cell knows that this cell has its target inside of it. So perforin is going to initially poke holes in the surface of the cell, and then granzymes that are being secreted also are going to go through those holes and chew up everything that's inside that cell. And it's a nice kind of restricted way of killing this cell instead of just like causing it to burst open and leak everything, where say there were a whole bunch of viruses in there, viruses get released and get to start infecting other cells. Instead, they're kind of restricted in that cell, granzymes go in and just chew up everything inside of it. So this response here, where the cytotoxic T cell is being activated by an antigen-presenting cell, usually also happening in the lymph node, this can be further activated by a T helper cell. If you go through that T helper 1 response we just mentioned, that response can include cytokines that further activate the cytotoxic T cell. Make it from just like a, novice assassin to being a trained 007 kind of assassin. Then it just goes surveying around the body, and of course, say you have infected by hepatitis that's affecting your liver. This cytotoxic T cell keep wandering around your body. It may go through your bloodstream and into your lungs, into your kidneys. Doesn't find any target there. Happens to pop out of your bloodstream into your liver. Then it starts finding the infected cell. So it secrete porphyrins and granzymes, kill the target cell, start sampling other cells until it comes across another infected cell. Kill it, then keep going, and it's just mass killing like that until it's run out of target cell. So, cytotoxic T cells, they'll specifically target certain host cells. Of course, bystander effect, the cell does get killed. That's your own cell if it's infected by a virus. It's not like that cell gets to survive. Viral infected are very common, and that's based because that infected host cell of yours is expressing viral proteins, like we know from when we covered viruses. Intracellular bacteria or protozoa that are hanging out inside the cell. Their pathogens can survive in the phagosomes, or maybe they've come out into the cytoplasm, but because they're intracellular, peptides from those will still be expressed on the surface. Cancerous cells, so a lot of those peptides, once the cell becomes cancerous, starts to look abnormal, and that's when it's going to get targeted. And then non-self human or other animal cells. This is what we'll go into more detail on Thursday. Graft versus host disease, like when you get an organ transplant. But in this case, it's usually because of the MHC being different than yours, rather than the peptides being presented being different. So, let's play a video to wrap up cytotoxic killing.
Each cytotoxic T cell produces a unique T cell receptor, which corresponds to a specific antigen. Each cytotoxic T cell also produces a receptor called CD8, which recognizes the MHC molecules of infected cells. When a cytotoxic T cell encounters an infected cell with a specific antigen bound in an MHC molecule, the cytotoxic T cell produces receptors for interleukin-2 and other cytokines, such as gamma interferon, IFN gamma. The cytotoxic T cell can then be activated by cytokines from activated helper T cells. Once a cytotoxic T cell becomes activated, it proliferates, forming a clone of cells specific to the same antigen. Some of these cytotoxic T cells differentiate into long-lived memory T cells, but most mature to attack and destroy infected cells. Cytotoxic T cells can destroy infected cells through multiple pathways. The best understood of these pathways is the perforin granzyme cytotoxic pathway. When the cytotoxic T cell encounters an infected cell with the target antigen bound in an MHC molecule, the T cell secretes two types of protein molecules, perforin and granzyme. The perforin forms a pore in the membrane of the target cell, allowing granzyme to enter the cell. The granzyme activates the enzymes inside the cell, leading to programmed cell death or apoptosis.
It's pretty awesome how they do that. And probably your cells actually, like your body, goes through producing cancer cells every day. It's just what gets past all your normal screening. You've probably heard in physio of like cell cycle checkpoints and things. But besides those, of course, still see some get through that stuff. That's where your immune cells, like cytotoxic T cells, come in. That's why you're not developing cancer every day. They're playing a big part in that because they're catching the cells that get passed all the